Thank you. It's my pleasure to introduce you to our two collaborators in crime. This morning, Professor Alan Boyd, I'm sure, needs no introduction to this audience. He is a former president of our faculty. He has made a faculty of pharmaceutical medicine known beyond our own faculty shores, even by appearing within the BMJ. He and his organisation is the major sponsor for today's meeting, and he is going to be collaborating in a conversation with Dr Ulrich Granzer, who's joined us from Germany today. Ulrich is the founder of Granzer Regulatory Consulting, and previous to that, he held senior positions at Glaxo, BASF Pharma and Knoll before joining Bayer as Vice President of Global Regulatory Affairs. Ulrich and Alan are going to have a conversation this morning to touch on the topics of regulatory sciences in clinical development. Thank you very much.
Thanks, Penny, very much. Now, I'm glad you're all sitting down because we're going to do something quite different. As Penny said, we're going to have a conversation, but we're not going to use PowerPoint. So it's going to be a proper conversation. Yeah, breaking the mold, that's good. So what Ulrich and I are going to do, Ulrich and I have worked together probably 15 plus years and doing various projects together across the world. We're each going to introduce very briefly what we do as a day job, because most of you know me as what I've done for the faculty, but I do actually have a day job as well. So that's quite important. And I'll talk a bit, we're just going to talk about that and what approach we take with regulatory strategy, and Ulrich will do the same. And then we're going to have, as Penny said, a conversation and ask each other questions about how we go about it for about half an hour. And then we'll open it up for questions, so you can ask us questions. So my day job, I've run Boyds Consultants now for about 15 years. My background, I've been in the industry 35 years, was a clinical pharmacologist, but I went through big pharma, ended up as global head of medical research at Zeneca. When they merged with Astra, I wasn't Swedish, so out I went. But I took totally the opposite direction and set up a startup spin-out company, and this was 20 years ago now, Arc Therapeutics. We had IP from University College London and a Finnish university in Kuopio, and we formed one of the first companies devoted to gene therapy. There was 3 of us, 2 ideas and no money, and people thought I was crazy. And the other thing I was going to do was do it all virtually. We grew that company, we raised money, venture capital, we floated on the stock market, and I developed the first gene therapy that went to the European Medicines Agency for approval, a product called Serapro for malignant glioma. This was the mid-70s. It didn't get approved, but there were many reasons behind that. But we showed it could be done, and that then paved the way for other things. Now, after that, and we didn't get Serapro approved, we were in the middle of the recession, couldn't raise any more money, so we closed the company down. But that's when I got into consultancy. And most of the investors and the financial people I'd been working with encouraged me to set up the consultancy to help them do due diligence and support their development. So where we started out was mainly working with small spin-out companies and a lot of universities. And I've continued to grow that over the last 15 years, and I've got a team of 25 people. We have clients from Sydney to San Francisco. Most of our work is advanced therapies, clearly because of what I've done with gene therapy, but we do all the other crazy things that are going on at the moment with endosomes and all that sort of stuff. Clearly a large part of that work is regulatory strategy and the activities, and that's what we'll be talking about. So that's me and my company and where I've come from, and I'll ask Ulrich to just describe where you've come from and what you do.
I can do so. Yep. Thank you very much, first of all, for the invitation. It's a great honor for me to being here, and I'm probably the only one, not a native speaker in English, so please be with me if I use words in different fashion than you usually are used to them. But overall, coming back to the point Alan is making, we have a rather similar history in that respect. I started my business life in a small company called Glaxor, just around the corner, and it became very, very big. I changed to a slightly smaller company called Knoll, where I was running regulatory affairs globally, and the first development we did was the first fully human antibody. We called it D2E7, a little bit like from outer space, but this thing became then Humira and became sort of successful, I've heard. After that, I was asked to work with Abbott, and they asked me to work in Chicago, and in addition to that, also in the rest of the world, as they called Europe. And the idea was I stay 1 week in Chicago and the other way a week somewhere in the world. And I tried that for a total of 5 weeks, where I was 6 times in Chicago at that time. And then I said, give me a place where I can stay. And then the then boss of Abbott, Miles White, said, oh, don't worry, Ulrich, I'll take the private jet or fly first class. On my way back home, I canceled the contract, went to Bayer, did my job there, and after I learned enough in big pharma, I started my own company, which is now based in Munich. And we are focusing on new drug development, drugs which are first in class quite often. In that respect, I also met Alan for the first time with ARC Therapeutics, where we tried to help getting it over the hurdle. We failed, as you have heard, but the next one, which we were involved in gene therapy, was a thing called Glybera, and with Glybera we got an approval. We therefore showed first with ARC and then with Glybera, that overall it is possible to get an approval for that, and this, at the end of the day, led to the increase in the number of companies founded on that basis, and also made it happen that gene therapy nowadays is really something which is becoming more and more important and more standard development. We are focusing in that respect on all what is necessary to bring new products into the clinic in the first place, and secondly then help companies going on developing the products up to approval. Then we step out. This year, for example, we brought in about 20 compounds into the clinic, and what we would like to discuss today is really what we are going to do in this respect. Thank you.
So, Ulrich, one question that we both get asked quite often is actually, in terms of scientific advice and pre-INDA meetings, what's the best approach to take for early development? I mean, do you go to see some national authorities here in the EU? Do you go to the EMEA first or the FDA? Or do them in parallel? What's your approach on that?
That's something we are getting really asked very often, I must say, because most companies think, and especially US companies, do think that we do have one agency in Europe, which is called EMA, and that's the European FDA. Then the first thing what we usually do is explain that the EMA is not something like the FDA, but this is an organization which is a secretariat to all the other agencies in the European Union. We have more than 40 of them. And then explain that the expertise is not with the EMA, but, and I hope not too many people of the EMA are around today, but that the expertise is with the local authorities, with the national authorities, MHRA, BfArM, MPA, PI, and so on and so forth. And then when we go for the very first time to an authority, we usually pick the authority which in that particular arena area has the best expertise in our view and in our knowledge. So we usually go to a little shopping tour of authorities, 2 or 3 or 4, depending on where we are. One might quite often be the British authority, the MHRA, where we go to. The other one, especially when it comes to advanced therapies, there is 2 leading authorities in the world. One is the authority in Silver Spring, CBER, of the FDA, and the other one, interestingly, is together with the MHRA, the Paul-Ehrlich Institute in Germany. So whenever gene therapies, whenever new products in the field of cell-based therapies, or also oligonucleotides or the like, or antibodies are being discussed.
Sorry, I'm just interrupting you for a second. Let me see your microphone.
Whatever you do, just do it. Can you hear me better now? Hey, good. Do I need to repeat everything, or is it? Okay, good. Okay, so we go and see those authorities, prepare a briefing book, and this briefing book preparation is the other important bit. Quite often when we have young companies, and Alan knows that as good as I do, young companies are focused on research. Then there comes this bloody thing which is called development. Development is something about guidelines, it's something about following a certain path, following a roadmap, and so on and so on. And for the very first time, quite often, the companies are forced to put that together for the very first time. That's usually the briefing book. We help them writing, and with that briefing book then and the questions they would like to ask, we go to the authorities, have the discussion, help them moderate the meeting, and then get out and have with that the first roadmap in place on how this development can go on. So coming back to your point, we usually go to national authorities first with this initial development thoughts, and then after that, and after we have clinical data and a bit more clinical data, then we go in addition to that, to the European Medicines Agency, and there to the Scientific Advice Working Party, which is the group coming from the national authorities, which are the experts in a panel where these developments can be discussed with.
Yeah, no, that's entirely my approach, and it's interesting, isn't it? I'm doing a lot of work, particularly with the academic groups. You know, when we suggest we've got to put a briefing book together, the usual initial response is, but I've got a paper in Nature, can't we just send them that? So it's taken into account. And when do you think, Aurics, the best time to go for these meetings?
Yeah, we usually have quite fierce discussions about that, because when we come into the place, we cost money, so they need to pay for us, and I know that Alan doesn't work for free entirely and completely either. So we are asking for a ticket, and that means that they need to swallow hard first, but then we explain why we should be going, because they need to really, for the first time, focus on how to develop a drug. And at the very first time point, when they start developing a drug, this is the first time point to go to an authority. So before spending big tickets in money, before spending a lot of money in CMC development, before spending a lot of money in classical toxicology, and especially before going into the clinical development, we seek scientific advice from the authorities. In other words, we ask the authorities whether they validate the program we are going to put together.
Yeah, and one question I get, because, you know, like you, I like to take companies and even the academic groups very early on. Mm-hmm. But a lot of them say, well, we haven't got any data, any clinical data yet. Aren't they going to think we're stupid? You know, so what's your minimum amount of data that you would go with? What are you really looking for?
That's a very important item, and at the same time an issue for many companies to go in early, because they exactly tell me, we don't have data, we can't discuss anything. A typical scientist's view of things in that respect, but not the view of a drug developer. So what we need as data is maybe a mode of action, an idea how a drug could be working, and maybe a couple of pharmacodynamic models. This could be cell-based models, but also, as we heard this morning, already quite a few animal experiments using skid mice or whatever might be a relevant species for this kind of testing. And with those data, putting together a plan on how to further develop, meaning what can be done in the toxicology part then to further see whether this is a dangerous way of developing things. And then, in addition, also showing a pathway to the first in human study, and we're going to probably come to the first in human studies in a couple of minutes. So the data necessary are two. The one is an idea on the proof of concept. It might, where the drug might work and how the drug might work. And the second part is an idea on how to go on. With its idea on how to go on, is usually the part where we step in and help them. So early on, very early on, before you spend the big tickets in money. And this is another important item here. Alan mentioned already that you need quite often investors in the beginning. Investors do want to not just pour money onto you and hope that it's working, they also want to know for what they pour the money to. And if you have a drug development plan which has been validated by a national authority in Europe or by the FDA, then you can build the ticket on how big the first parts of the milestones are. You can tell this your investors, and then usually you have a much better chance to raise that money, which means the regulatory authority interactions in the very beginning are the first milestone to get money into the company. I mean real money, not funding money from business angels.
And the other thing a lot of people forget is the manufacturing side and the CMC, because, you know, as clinicians and things, well, surely that just turns up, doesn't it? No, it doesn't. But you know, a lot of the times we're taking people, we do lay out the proposed plans for how the therapies are going to be made. I mean, now with the cell therapies and the gene therapies and advanced therapies, we more or less know the process. But what we've got to do then is fill in the gaps. So one important thing, one of the earlier questions that we often put, are things like release criteria for the medicines as they're being made. You know, what is the batch releases? Because clearly in the potency assay, it's quite interesting. In going for these meetings, particularly with the FDA, they will often, when you go in, you see good morning. The next question is, what's your potency assay? Because that's so important to me.
Especially at CBER, and there is a lady called Elena Gubina. She is the potency assay lady at the FDA. She wants potency assays for everything. Yeah? You talk to her about a cake, she asks for potency assays. You talk to her about tea, she asks for potency assays. And that makes life a bit more complicated quite often. Another important aspect in that respect is you must not go into clinical trials without GMP material. Quite often, we need to explain what that G stands for, and then this M stands for, and that P stands for, and especially in young biotechs. And then they say, okay, we can make that drug. Full stop. And I usually ask, what do you mean by you can make that drug? And then, for example, the nice story comes, oh, I have this roller bottle in my office, and every single day I roll it, and then half a liter of fermentation process in there, and we get the drug. And then we need to slowly shift them into real world, not evidence, but real world work. And that also is quite a big task. And I'm sure you're anticipating the same thing when you're talking to many fresh companies.
I can't name the company, but what I'm working with at the moment, they've done just that, developed a process on the bench. And we were talking about where we're going for manufacturing, and the CEO, who actually comes from telecoms, so he can't really understand the urgency. He did ask me, why does a 3-month toxicology study take 3 months? Can't we shave some time off it? And he fell off his chair when I said, well, actually, it takes 6 months. But anyway, they're like that. But his idea was, look, I've found a manufacturer. I'm going to put the 2 research guys. They can be trained in this GMP stuff in about 2 weeks and then make it. Is that all right?
Absolutely. That's what I'm hearing as well. I also heard from an investor a nice story about a carcinogenicity study. I told him it's a 2-year study in mice, and then in addition to that, all in all, the histopathology. And so we are going to calculate usually almost 3 years for that. And then he said also to me, this has to be faster. And I said, what kind of idea do you have? I'll give them more drug, he meant. So if you pour that into the mice in 2 years, why don't you do that in 4 weeks? And I said, this is a very good idea to develop a rat poison, but not a drug. And so we had a funny conversation at the end. We had a beer afterwards, and then he didn't understand it slowly, but it needs this time of education.
Yeah. And what about taking experts and key opinion leaders with you to those meetings? I mean, one person I never take is the CEO. I don't know about you.
I usually try to avoid CEOs as well, and because they are. How many CEOs are in the room? No, the simple issue is that they are usually used to be the big elephant in the room. This is part 1. Part 2, we have no fallback position anymore if the CEO is around. So if an authority, for example, says it would be good to do this other trial in addition, as a moderator consultant, I always can say, yeah, we take that into consideration and we'll look at it. If the CEO is there, and I've seen that, he just says, oh yeah, good idea, we'll do it. 10 million pounds later, and a couple of harsh discussions with the investors later, he is all secured from the idea going to the next authority. And these kind of stories, I usually tell them to leave them out. KOLs is the other group. as long as they are, and again I need to look around, as long as one can control them,
Yes.
it's okay. So we do a little session prior to the meeting and train them, educate them on what is possible, what can be done, and what shall not be done. For example, we had a funny or nice question. This was at the FDA, and we had a meeting. This was a pre-IND meeting, and we had a KOL with us, and he said, now that we have discussed all of that, I have a question for you as well. What else can we do? And this was when we discussed clinical trials. And one guy at the FDA had the idea that another clinical trial with about 300 patients would be a good idea. So the KOL has just brought over the company a new trial of about 15 million pounds. He just had one question. And so it is really important to control. The other thing which is also very important to control is when he or she starts to lecture too harshly, because he or she thinks that the guys on the other side are not real scientists but something else. And then they give them a nice lecture from the lantern straight down. And that's also not something very well taken by regulatory authorities. So you need to really look for the character of the person and train them.
And one final thing about the meetings, the FDA now will offer very reluctantly face-to-face meetings. Mm-hmm. Most of the work and interactions we have with FDA are by teleconference. Mm-hmm. And even then, they're getting just written feedback meetings. Do you think we're losing something by not having those face-to-face meetings?
Face-to-face meetings are something companies really want, especially when they're young. They want to see the FDA, and they quite often they are for the first time in Silver Spring and for the first time at the buildings there, and so they like that. The second part is they think that only a face-to-face interaction really brings across what they are going to discuss. What we have learned, especially in pre-IND meetings, so in first interaction meetings, is that a telecon might be even better than a face-to-face meeting. That sounds strange in the beginning, a little bit. But if you have a good briefing book, and if you have then a question which is a bit divergent and needs discussion, then the FDA has the chance to go into mute on the telecon and discuss that internally and give you an answer. If you are in a face-to-face meeting, that never happens. Then you don't get an answer. So in early meetings, it's quite often even better to have a telecon and get that. And if the briefing book is being written well, and if the telecon is moderated well, which is also something special, and we could talk hours about that, then you get not only the same result, but quite often a better result.
Okay, let's switch topic then. Orphan drugs.
Yeah, okay, orphan drugs. We both have some kind of an affinity to orphan drugs and done quite a few ones overall, and very often raised and asked question is, orphans is always great, that's part one, and first of all, the question would be, why is orphans always great by getting an orphan drug designation? The second question would then be, at which time point should that be done?
Yeah. As you say, I think we've done probably about 40 plus orphan drugs now. We've done a lot of work for American companies coming into Europe. So if you look on the EMA website, you'll see that Boyds Consultants holds about 12 orphan drug designations. I should say they're not ours. They're just parked with us and moved over. But the approach, the question is often, should we go to FDA first or EMA? Mm-hmm. It doesn't really matter, I don't think. We tend to go with, because clearly with American clients, what they want to do is to get into Europe and go to the EMA. So probably more often than not, we start with EMA, because also it's a longer process and it's defined. You have to submit things at certain dates. You might have a submission meeting, so that extends it. I mean, the whole process takes a good 6 months, if not more. Okay, it's timed and calendared, but you have to work that in and take that into account. The FDA, it's less structured, very similar information that you put in, so it really is a cut and paste job between the submission documents. Clearly the prevalence, or now more recently with the EMA, it's the incidence data they're looking at. And of course for Europe, we have to produce that with European data for the orphan drug, and also for the FDA with their figures. So that's really what we do. And I do encourage companies to go for both very closely together. I had one instance where there was a company that we filed the EMA, for the Orphan for the MEA, and they said, well, we'll hold off with the FDA. The problem was this company had got quite a lot of publications out there, and once it was announced that they got orphan drug with the MA, a group, an independent group, pulled together all their papers and then filed an orphan drug application in their name based on these papers, which sort of, you know, clearly the company could have filed again and things like that, but it's, those sort of things do happen. So that's why I like to go really with both, more or less simultaneously. And clearly, you know, there are advantages, you know, as well as the, you know, the incentives that you get. But people often say, what's the point of getting it at this stage? But actually, having an orphan drug designation is very good for fundraising.
That's the most important
Yes.
point. I think we should be saying, because a lot of investment groups think that an orphan drug designation already is a kind of proof of concept or a kind of proof that the drug might work and that there is a good business fortune for it. Maybe you can say something about that as well.
Yes. Well, I mean, they are, you know, in terms of having that, they do, you know, help you raise money and things like that. Though it's interesting, you know, you raise things about issues. Sometimes I find the orphan drug groups particularly frustrating, with particularly the way that they look at the population and the disease. And I've got a couple of examples. I was working with a Finnish company who were developing a growth factor, a gene therapy for the treatment of lymphedema for patients who'd had breast cancer and then developed lymphedema. And what the technology was, because they've lost lymph nodes, they're transplanting lymph nodes from the inguinal or femoral region, putting them in the axilla and then injecting the gene therapy around it and to grow a new vascular system. Now, there's a couple of other conditions that, mainly melanoma, and that also cause lymphedema, but in the legs. And clearly, you're putting a growth factor in, and the idea was you'd wait for a couple of years after the breast cancer had been treated to then do this, because the concern was you're giving a growth factor, it's a VEGF-C gene. Would you then stimulate the cancer there somewhere? And so we went purely with this indication in breast cancer. And it met the criteria for orphan designation. But the orphan drug group in Europe, and particularly the medical review, said, look, you can get lymphedema in ovarian cancer, in melanoma, so therefore you're outside the orphan conditions. And I argued strongly, I said, nobody in their right mind, because of having ovarian cancer and melanoma, would you actually give a growth factor to? They said, well, they might do, so therefore we're not giving it you. Which is really quite surprising that we couldn't get. The Americans granted it, no problems. So different approach really. But sometimes I do get surprises. Working with Spark Therapeutics, with Luxturna, this is the gene therapy that's recently been approved for inherited eye disease. This was an interesting debate, because when the orphan drug went in, probably about 8 to 9 years ago originally, it was just given its clinical diagnosis, Leber's disease or retinitis pigmentosa. When the gene therapy is specifically for a gene mutation, the mutation of the RPE65 gene at the back of the eye. And when the medical group, and in fact the CHMP reviewed it, they said this drug should be indicated to a specific gene mutation and not a disease name. So this is what, the first sort of debate in the world, that we're now going to have a gene therapy for a gene mutation. But the orphan drug group fought back hard. They said, no, we've granted it for the name, not the mutation. And it took us, and Spark have talked about this, probably took us nearly a year to resolve that. In the end, the orphan drug group saw light, which is rather strange, isn't it? So now when we're putting in orphan drugs for gene therapies, we actually are thinking a lot more about actually naming the mutation for the treatment of the RPE65 gene, not for the name of it. So there's some surprises there.
Which also has a consequence, because if you name the gene, even broader diseases could become orphans.
Yes.
As you all know, pricing in orphans is a specialty. I've recently had a discussion with this group at the EMA and told them in the future we are coming for orphan type 2 diabetes, just making it up, or high blood pressure or something like this, because we just use one gene, and based on that we shrink down the group to a very small population, and then we have type 2 diabetes therapy for $500,000 a year. They were a little bit reluctant in thinking about that, but then they started thinking of that, and then we are coming back a little bit to giving a broad indication and thinking about the condition itself instead of just going into a little single mutation on the situation, which would then trigger a completely new way of looking into diseases as such. So that's the other discussion we're facing right now as well.
Yes, yeah. I mean, it's clear we are, want a better word, stressing the regulators with these new things that are coming out.
Yeah.
It's, you know, they're having as much difficulty as we are, actually, and sorting that out, and clarifying this. But, you know, these are the things that are being decided now, paving the way for the future with what's coming out. So, what about clinical study designs? The amount of time I spend on that, and the ideas that, you know, you need 2 randomized, well-controlled trials to get your approval. And,
I think that fits extremely nicely into the discussion we just had.
yeah.
We have seen orphan drug approvals with single-arm trials. We have seen orphan drug approvals with phase 1b trials, meaning trials which were the very first ones. We have also seen orphan drug approvals where we had a total of 11 patients in the entire database. We had one case where we had 1 patient in phase 1, 2 patients in phase 2, and then came the large pivotal trial, 8 patients. This led to an approval, by the way. So we are really moving away from classical RCTs to pivotal trials and a lot of other trials in the beginning.
Yes.
Maybe you can go into also your thoughts about single-arm trials and
Yes.
historic controls in that
And
respect.
historic controls. I mean, well, single-arm trials, you know, are becoming much more accepted by the regulators. I've been doing some work with a gene therapy for bladder cancer, end-stage bladder cancer, and the next stage in that treatment pathway is cystectomy. So you can't actually do in that patient population a randomized controlled trial because it's not ethical. And we knew we wanted to have a single-arm study, but I went to the FDA and he said, look, I want a single-arm study, and the 2 medics said, oh, just a minute, you need 2 randomized controlled trials. And we had a big debate, and in the end I put my pen down, and there was a senior medic there, and I said, look, this is very difficult. I got experts. I did take a couple of urologists experts with me, and they said, this is what we can do. I said, what can we do here? And if you were us, what would you do? And the answer was, we don't know. And what we decided to do then, we actually held a small symposium between the academic group, the Urologists Oncology Society in the States, the company, and the FDA. And what we worked out was that we'd do a single-arm study. There had been another product approved, Valrubicin, that had got an 18% complete response rate in this group of patients at 6 months. And out of that meeting came, we'd done a phase 1 study where we'd seen a 38% response at 12 months. And so from that, we worked the endpoint out that we would do a single-arm study, and if we got a complete response rate of greater than 25%, we'd get approval. We've recently submitted the BLA to the FDA. You can guess what the result is. It's probably greater than 25%, otherwise we wouldn't have sent it in. But that's happening. So a good example, really, the single study.
Yeah, and I think you mentioned the most important item. If you go for a historic control and if you go for a single-arm trial, the effect size you're looking at needs to be large. The larger the better, but so large that it couldn't be reached by chance based on historic data. And one needs to discuss with the regulators, coming back to the first item we were discussing today, we need to discuss with regulators then exactly this item and exactly getting to the point that we reach agreement on the effect size, the endpoint, primary endpoint, the secondary endpoints, because they should be consistent with what you see in the primary endpoint and all of these items, and then eventually such a trial or such a development program, which consists sometimes of just one single arm trial, might be a possibility.
Yeah. The other thing, obviously we're doing a lot of now, are natural history studies. and run in studies into these rare disease areas, because again, one of the problems I find is when you're trying to choose an endpoint for the study and the primary endpoint, because a lot of these rare diseases, because there's no treatments, you don't know what to measure, what the outcome is. And I think Glybera actually was a good example, because, do you want to explain that one?
Yeah, actually Glybera was the very first gene therapy approved. It's LPLD deficiency, so there's an enzyme defect which usually leads to breakdown of the chylomicrons in the body, and this enzyme is then being given via the gene therapy, so it's de facto an enzyme replacement therapy which is being done there. In the beginning, the idea was to just measure lipids in the blood, but just breaking down chylomicrons doesn't reduce the lipids overall. So this endpoint was an interesting one, but not doable. Unfortunately, the clinical trial had already that as an endpoint. So we
that
were
was my fault, actually.
Oh, good. Good to hear that. He never told me that, by the way, when we were in the process, that this was his idea. But okay. So then we had to invent, while we were in the final steps of registration, the right endpoint. And in that respect, I should also make a remark that in the first 10 orphan drug approvals I was involved in, always the primary endpoint was wrong. It was always the endpoint which showed the least from the disease and the improvement of the drug. So we always had to look into something else and find, during the process of filing, a new one or a better one. Most often one of the secondaries, but I can also remember one case where we have had a tertiary or experimental endpoint which made it then. In this case, we found that the amount of chylomicrons in the blood was the right endpoint, and we had at the end then 3 patients in total in the entire database which had the right endpoint, the right times to measure, and the right visibility. So we got an approval, and you can read that in the European Public Assessment Report, where we had a total of 3 patients with all the right data, which led to the first approval of a gene therapy in the European Union. Difficult to get below this number when you are developing a drug.
Now, we've obviously talked a lot, time's getting on. Can I open it up for, are there any questions from the audience? There's 1, 2 hands going up. Gillis, that's you.
So, both of you, with your experience of both regulatory authorities, EMA and FDA, could you compare and contrast the quality of the scientific advice which you have received for your small early stage companies, particularly pertaining to oncology, which is 50% of biotech activity, as we know? Could you compare and contrast the 2 agencies in a few short sentences? Is that possible?
Yes. With oncology, I find over the years they do, the regulators do change in their approach and the sort of advice you get. I think at the moment, from my recent experience, the FDA is probably giving better advice at the moment than the EMEA. But I think that's probably because of, and I hate to mention the B word, Brexit, and all the changes that have gone on. And there's been a lot of people leave there, so there it is. Opposite oncology, Gillis, there's always this debate, is it progression-free survival or is it overall survival? I think at the moment progression-free survival, and interest yours, is more favoured in the States than it is here, in Europe, shall I say, but any
yeah, actually, the latest discussions we had in early oncology programs, we quite often, we usually go to, I should say, to the FDA as well as to the Europeans, and then we select a couple of agencies where we think that we get the best feedback. One is usually the MHRA, but then we usually also go to the Swedish authorities and sometimes to the Germans in addition as well, so that we get a good overall feedback. What we see is that the FDA is more open to earlier readouts, overall response rate, responses as such, PFS, and only in the late stage overall survival, whereas the Europeans are more conservative in that respect. What we also learned a bit of the hard way is to not go to the EMA directly, because there sometimes it happens that somebody from the scientific advice working party who takes up the case and is the kind of rapporteur for this case is not an oncologist, and I should almost say has no clue what he's talking about. And then you get, I think the right word in English is an interesting advice. And Then we have to go for a second advice or for a repeat advice, and there the FDA is extremely helpful, because if we get a good advice there, then the company can at least go on and develop. And we have one place to go back to and even file with good data. We recently, for example, filed with a basket trial where one basket looked good, phase 1B. FDA agreed to that. We went to the Europeans as well, and the Europeans said, okay, that's great information, do another trial. FDA said, send the BLA. So that's really the difference between the two right now.
On the right.
I'll shout it out a bit louder then.
Thank you.
I've noticed this talk has been basically US, EU perspectives. Clearly Japan is the world's second biggest market.
Yep.
Would you like to pass a few comments about strategies for Japan and how that fits into a global strategy?
Yeah. The approach really is to, because most of our, the clients, and I agree with you, Japan's a big market there. We tend to start with Europe and the US. Remember, these are small companies a lot of the time we're working with, and they don't have the funds to do, everywhere at once. So it has to be staged as part of the strategy. So probably the next place we'd go to, once we'd got work going in either of those territories, is then to think about the Japanese authorities and to go to that. And we've done work recently with Canada, Australia, Singapore, those places as well. So.
Where in that respect, Australia, Canada, also Singapore is a bit more European in that respect, or US, or more European than US, and it's a bit easier in that respect. Whereas when you go to Japan, you have to follow a couple of rituals which are just the way how things are being done in Japan. One thing, for example, in order to get a good interaction with the authorities is that you do an investigator-initiated trial in the beginning, 5 patients, 6 patients, 8 patients, something along those lines, so that the drug has been used in a Japanese population, as they call it. Always only 3 patients or 5 patients, depending on how many it are. But with that, you then can have your initial discussion with the Japanese authorities. And there's one other thing. I've been there a couple of times, but I'm not going anymore because I'm the long nose sitting there having a translator with me, and I'm not helpful in these meetings, to say the least. So we have a partner on the ground there who does that for us, with us, and with our briefing book, and they speak proper English so that we can communicate with them, and they then can, in Japanese, communicate with the authorities.
I'm going to just take slight exception to this advice, because there are occasions where the Japanese and indeed the Asiatic races have higher incidences of certain types of cancer than in our part of the world, for example. So actually getting early advice from the PMDA is extremely important, because the market is actually not the Western world, it's the Asian world. In addition, we kind of overlook the fact that Asia is a major source of investment monies right now, and so it isn't necessary to raise money in Europe or America. Actually, you can raise money in Singapore, in Japan, in China, and why not? Particularly where you have an asset that may have more value in that territory than in your own. So I'd just like to share my experience.
No, actually, that was the more generalized approach. We, for example, are right now in the development of gastric cancer products, a couple of those. And there we have started with the work in Japan and in China, not in the Western world. And we have a couple of other vaccinations, for example, for diseases which are in this area much more prevalent than they are here. There we go straight. I think what the two of us gave as a response was more the general attitude towards a European or a US biotech company. They usually go to their home market first, and we help them getting that first done, and then jump over the Atlantic, and before we become Pacific, we normally have a couple of hard data.
It's obviously because I lived in the Pacific too long. Further questions? Two? Maybe the fifth from the front row first. Thank you.
Thank you very much. Can you hear me? So I just wanted to follow up. You know, you've given the perspective here in terms of scientific advice in Europe from a regulatory perspective solely, but you know, more and more we see joint advice also being given now with the HTA bodies. So could you just comment on that as well? At what point do you actually in Europe bring the HTA bodies in as well?
Yeah, I like to start thinking about it really once we've got some phase 2 data. Again, though, it becomes a bit blurred because with a lot of the advanced therapies, we're not doing phase 1, 2 and 3 in that sort of sequence. You know, as Ulrich said, we've, you know, a lot of these gene therapies, like, you know, for instance, Serapro, we went from start to finish with, you know, with a phase 1 and then the pivotal study of 400 patients in about 4 years. So it's moving very fast. So I think really probably is when you're getting into your pivotal study. I know the Americans don't like the word pivotal, the FDA doesn't like that word anymore, but that's probably when we start thinking about HTA assessments at that time, at least starting the discussions.
But an assessment of the real world market, and this is where real world evidence may be of particular relevance, is this question of what is the value of an asset in the real clinical practice, because that is what sways an HTA assessment, not an artificial endpoint.
Yes. Actually, what we normally do is when we, for the first time, work with a company and they first develop their indication, we do a broad assessment on what is there already, meaning what kind of drugs are there, what kind of treatment and therapies are there, and what is the value of these therapies. And only after that, sometimes we stop the project at that point already, because when you develop this great new NSAID, for example, it becomes very difficult to make it a good thing or think payers pay anything for. So we stop it there. But in other cases where we then see that there is a good niche, for example, which we can then expand on, we start from there onwards also to look into the endpoints, and from the proof of concept study onwards, we let these endpoints run with it. That's not necessarily always the primary endpoint, but secondary endpoints which have a meaning for the HTA bodies are included usually at a time point, phase 2B at the latest, and then in the, and I'm not saying the P word, in this final study in addition, absolutely as well.
I think that's the importance, the endpoint issue there.
Thank you. There's a question 3 up from the front.
Yeah, I think you touched on this briefly, getting regulatory approval in China. Could you talk a little bit about that? Because it's mostly UK, EU and Japan that it's focused on.
Yeah. China's an interesting place. In fact, Ulrich and I were discussing it first thing this morning. Do you want to relay the conversation we had?
Yeah, maybe I can do so. The Chinese market becomes more and more important, and there's also a lot of Chinese companies, especially in the biosimilars world, which are approaching the rest of the world, as they call the rest of the world as well. What we right now see is that the legislation in China has been adapted in the last couple of years, 2 to 3 years, 4 years, to make a policy which one could call China first. When we approached the Chinese authorities, they've just renamed, by the way, and I've forgotten the new name. It was the SFDA, and now it's called something different. The idea behind that is that if a drug is approved in China first, you get data protection in China. If the drug is not approved in China first, you don't get data protection. So the idea behind that is you develop your drug in China first, or you develop your drug in China, the first approval is being done in China, then you get 5 or 7 years of data protection or data exclusivity in China. If you file first in the United States, for example, and you file a year later in China, this 1 year of approval time is subtracted from the Chinese market. The idea behind all of that is that drugs should be developed in China and approved in China first and in the Chinese population. That's part 1 of the game. Part 2 of the game is we right now have a couple of global programs up and running where we have a big chunk of the treatment arms running in China. So we do global trials where Chinese patients are part of the overall clinical trial, and we do 2 kinds of assessments of that. We do an overall global intent to treat assessment, but in addition to that, we make sure that the single ethnicities are big enough to build strata, which then could be evaluated and assessed as well. The idea behind all of that is that we build one real global dossier with the specificities for Europe, the United States on the one hand side, and China and Japan on the other hand side, and then file synchronously, because that is the only chance to get the data protection right in China as well as in the United States as well as in Europe. So that's currently the idea, and for that purpose we also work with a Chinese company on the ground in China which does this kind of assessment evaluations with that. There's also one group which is a bit easier to access in that respect. It's the Harvard Medical School. There's a small group which does clinical trials, large clinical trials worldwide. And this group is headed by a lady called Yuan Di, and then she has the great name Halvorsen as her name, because she's married to a Norwegian. She speaks an extremely good English, but in addition to that, a very good Chinese, and she can directly negotiate things and you can discuss it with her. So there are a couple of groups now coming up which take that up, and we just do it in a way that we really develop it broadly, as broad as possible.
One final question, and then we'll bring this session to a close.
The regulators, both in FDA and Europe, are very clear that for orphan drugs and orphan indications, they accept new models, not two pivotal phase 3 trials. You alluded to that. They accept one phase 3 trial and another model, or sometimes both of them are not. But the trade-off is that they want more intense post-market.
Yes.
Yeah.
And usually is happening by way of registries, most of the time. My question to you is, what's the difference in the regulation or regulatory license for the, is it a condition of the license? And if the result, because usually the problem is that few patients in clinical trials are also means few patients in the real world, and the trial might not achieve its milestones and so on. So what's the regulatory commitment, and are they flexible when you can't meet the targets and so on?
Yeah, well, I mean, the Glybera, the first gene therapy to be approved, was a conditional approval. And they had to form a registry and they had to follow patients up. Unfortunately, there weren't many treated, and that was a problem for it. In fact, you know, it subsequently it's not available. The other gene therapies that have come out, I think, have all got full approvals. I think.
But with a commitment to registry.
Yes.
All of them.
But they've all got registries. But
Yeah.
the registry is not for efficacy. Well, in fact, it is really, because it's primarily been set up for safety. But one of the problems we have with gene therapy, of course, is, you know, they're single doses, where particularly you're going into the eyes and the CNS. The duration looks to be very good, but the regulators are still looking at that in terms of the registry studies. So, yeah, in fact, I'd be very surprised these days if a study doesn't get approved without some kind of registry and follow-up.
In that respect, just one further remark to that. If the result of this additional trial is not that good, so far not a single drug has been removed from the market. So the results will have to be presented to the regulators, but usually they are not leading to withdrawal of the drug. They may lead to further trials and further evaluations, but not to taking it off.
Okay.
And thank you very much indeed, Alan and Ulrich, for a lively session this morning.